This presentation is on renal function, Chapter 3 of the reference book, Urinalysis and Body Fluids, 6th edition by Susan King Strassinger and Marjorie Schwab Di Lorenzo. The audience for this presentation is students in a medical laboratory technician or medical laboratory scientist program. This presentation includes a lecture, definitions of terms, and some questions. My name is Jean Riddell, and I hope that you find this presentation useful in your studies. This slide lists key terms that you should understand the meaning of as a laboratory technician or laboratory scientist. These terms are active transport, afferent arteriole, aldosterone, antidiuretic hormone or ADH, beta-2 microglobulin, collecting duct, countercurrent mechanism, creatinine, cystatin C, distal convoluted tubule, efferent arteriole, free water clearance, glomerular filtration barrier, glomerular filtration rate, GFR, glomerulus, juxtamedular apparatus, loops of Henle. Additional key terms: maximal reabsorption capacity, nephron, osmolality, passive transport, peritubular capillaries, podocytes, proximal convoluted tubule, renal plasma flow, renal threshold, renin, renin-angiotensin-aldosterone system, shield of negativity, titrable acidity, tubular reabsorption, tubular secretion, vasa recta. Renal anatomy and physiology. The structural and functional unit of the kidney is the nephron. Each kidney has more than 1.5 million nephrons. There are two types of nephrons, the cortical and juxtamedullary nephrons. Cortical nephrons. 85% of the nephrons in the kidney are cortical nephrons. These nephrons are located in the outer layer of the kidney called the cortex. These nephrons function to remove waste and absorb nutrients. Juxtamedullary nephrons. The second type of nephron is the juxtamedullary nephron, which makes up 15% of the nephrons. These nephrons have longer loops of Henle that descend deep into the medullary region. The function of these nephrons is to concentrate the urine. The renal functions, renal blood flow, glomerular filtration, tubular reabsorption, and tubular secretion allow the body to clear waste products selectively from the blood and simultaneously to maintain the body's essential water and electrolyte balances. Renal blood flow. The kidneys are highly vascularized and receive 25% of all blood from each pump of the heart. The blood enters the kidney through the renal artery. The arteries decrease in size till they become afferent arterioles before entering the nephron. Inside the Bowman's capsule, the arteriole is called the glomerulus. As it exits the capsule, it is called the efferent arteriole. Then becomes the peritubular capillaries and the vasa recta. As the veins increase in size, they become the renal vein as they exit the kidney. Make sure that you don't mix up the afferent and efferent arterioles because they sound so similar. Renal blood flow. The peritubular capillaries wind around the proximal and distal convoluted tubules, allowing for reabsorption of vital nutrients from the proximal convoluted tubule and adjustment of urine concentration around the distal convoluted tubules. The vasa recta is the peritubular capillaries that winds around the loops of Henle of juxtamedullary nephrons. These are responsible for the kidney's ability to concentrate urine. An osmotic gradient is maintained in the medullary region so that water can be reabsorbed and the urine concentrated. If you need to better understand this very complicated process, I recommend watching Armando Hasurigan's physiology videos, Bowman Science Urinary System videos on YouTube. Glomerular filtration. Glomerular filtration occurs in the glomerulus and is reliant on hydrostatic pressure differentials between the blood in the glomerulus and the filtrate in the Bowman's capsule. The smaller efferent arteriole maintains consistency of glomerular capillary pressure and renal blood flow within the glomerulus. The glomerulus is made up of 8 capillary loops whose walls create glomerular filtration barrier. There are several factors that affect the actual filtration process, including the cellular structure of the capillary walls, the Bowman's capsule, oncotic pressure, and hydrostatic pressure, as well as the feedback mechanisms of the renin-angiotensin-aldosterone systems. The glomerular filtration barrier is made up of the capillary wall membrane, the basement membrane or basal lamina, and the visceral epithelium of the Bowman's capsule. The capillary walls are fenestrated and allow small molecules to move through, but prevent larger molecules and cells from passing through them. Cells called podocytes on the inner layer of the Bowman's capsule cover some of the fenestrations, further preventing larger substances from passing through into the filtrate. There is further a shield of negativity which prevents positively charged substances such as albumin from passing through the membrane. The pressure in the glomerulus is caused by hydrostatic pressure and oncotic pressure. The hydrostatic pressure is caused by the smaller efferent arteriole and enhanced capillary filtration of the glomerular filtration barrier. Oncotic pressure is caused by the ultrafiltrate plasma proteins in the glomerulus. The juxtamedular apparatus senses pressure and concentrations, causing the afferent and efferent arterioles to change opening size to maintain a constant pressure within the glomerulus. The renin-angiotensin-aldosterone system regulates blood flow to and into the glomerulus. The juxtamedular apparatus consists of juxtamedular cells, macula densa cells of the distal convoluted tubules. Blood pressure and sodium levels affect this system. Renin-angiotensin-aldosterone system. When the plasma sodium level decreases, this causes the kidneys to lose water to create balance, but this decreases blood pressure. This is sensed by the macula densa cells, causing the juxtamedular cells to secrete a substance called renin. When renin reacts with angiotensinogen, it converts it to angiotensin 1. When angiotensin 1 reaches the lung, it encounters angiotensin converting enzyme, or ACE, and is converted to angiotensin 2. Angiotensin 2 causes vasodilation of the afferent arterioles and constriction of the efferent arterioles, which stimulates the reabsorption of sodium and water in the proximal convoluted tubules, which then triggers the release of sodium-retaining hormone aldosterone from the adrenal cortex and antidiuretic hormone from the hypothalamus. As the sodium and blood pressure increase, the level of renin decreases due to a negative feedback system. Ultrafiltrate. The ultrafiltrate is created by a non-selective filtration of plasma, making it a plasma ultrafiltrate. The kidney filters 120 milliliters per minute. The differences are that the filtrate does not have proteins, protein-bound substances, or cells. The specific gravity should be about 1.010. Tubular reabsorption. The body cannot afford to lose 120 milliliters every minute. Therefore, the body must have reabsorption of parts of the ultrafiltrate. Reabsorption is when substances that were originally filtered through the glomerulus into the filtrate are reabsorbed into the blood. This occurs in the peritubular capillaries surrounding the proximal convoluted tubules. Tubular reabsorption mechanisms. There are two mechanisms of tubular reabsorption, active and passive transport. In active transport, a protein carrier combines with the substance to reabsorb it across the membrane of the proximal convoluted tubule. These substances include glucose, amino acids, and salts. chloride in the ascending loop, and sodium in the distal convoluted tubule. Passive transport is when molecules move across a membrane as a result of differences in concentrations or electrical potential on the other side of the membrane. Water is reabsorbed by passive transport on all parts of the nephron except the ascending loop, which is not permeable to water. Urea is passively reabsorbed in the proximal convoluted tubule as well as the ascending loop of Henle. Passive reabsorption of sodium accompanies the active transport of chloride in the ascending loop. If the concentration of a substance exceeds the maximal reabsorption capacity of the tubules, then the substance begins appearing in the urine. The plasma concentration at which the active transport stops is called the renal threshold. The renal threshold for glucose is 160 to 180 milligrams per deciliter. If the plasma concentration exceeds this amount, then glucose will be present in the urine. If a person with glucose in their urine does not have a high plasma glucose level, then this is due to tubular damage and not diabetes. Renal concentration of the filtrate into urine begins in the descending and ascending loops of Henle, where the filtrate is exposed to the high osmotic gradient of the renal medulla. Water is reabsorbed in the descending loop. There is selective reabsorption process is called the counter current mechanism and serves to maintain the osmotic gradient of the medulla. The sodium and chloride leaving the filtrate in the ascending loop prevent dilution of the medullary interstitium by the water reabsorbed from the descending loop. Maintenance of this osmotic gradient is essential for the final concentration of the filtrate when it reaches the collecting duct. The concentration of the filtrate leaving the ascending loop is quite low owing to the reabsorption of salt and not water in the part of the tubule. Reabsorption of sodium continues in the distal convoluted tubules, but is now under the control of the hormone aldosterone, which regulates reabsorption in response to the body's need for sodium. Collecting duct concentration. The final concentration of urine happens in the furthest part of the distal convoluted tubule in the collecting duct when water is reabsorbed. This reabsorption depends on the osmotic gradient in the medulla and the amount of antidiuretic hormone. The ADH controls whether the walls of the tubule or duct are permeable or impermeable to water. If you have a high level of hydration, you have less ADH and more urine due to less reabsorption. When you are dehydrated, you have higher levels of ADH and you reabsorb more water, leading to less urine that is more concentrated. Tubular secretion. Tubular secretion is the opposite of tubular reabsorption. This is when substances in the blood are secreted into the filtrate. This serves two functions. One is to remove waste that aren't filtered by the glomerulus, and the other is to maintain acid-base balance through the secretion of hydrogen ions. Acid-base balance. To maintain acid-base balance, the blood must buffer and eliminate the excess acid formed by dietary intake and body metabolism. Bicarbonate is filtered readily through the glomerulus and then reabsorbed into the bloodstream. The secretion of hydrogen ions into the filtrate causes 100% of bicarbonate to be reabsorbed in the proximal convoluted tubule. The complicated process and chemical reactions occur in the filtrate, renal tubular cells, and the plasma. This process works to maintain the blood in a tight range of 7.35 to 7.45. The process is fairly complicated, but I found a video by Alia Medical Media called Acid-Base Balance Animation that shows the process nicely. And you can see the link right here on the slide, and I will try to add this link in the comments section. Renal function tests include glomerular filtration rate or clearance tests for creatinine, beta-2 macroglobulin, and cystatin C. Glomerular filtration tests A glomerular filtration test must measure a substance that is filtered through the glomerulus and is not secreted or reabsorbed in the tubules. The substance measured must be stable in the 24-hour urine container, readily available in the body, and have a consistent level in the body. We are measuring how much blood is filtered through the glomerulus. When this level is decreased, it indicates kidney disease or damage. Clearance tests performed in the laboratory include the endogenous tests creatinine, beta-2 macroglobulin, and cystatin C. These substances are normally found in the body and filtered through the glomerulus. The exogenous test utilizes radionucleotides, which must be given to the patient and then measured in the urine. The endogenous tests are the ones we perform most often. Creatinine clearance test. The creatinine clearance is one of the most common glomerular filtration tests performed. Creatinine is a waste product of muscle metabolism. Some disadvantages are that it is secreted in the tubules as blood levels rise. The drug gentamicin, cephalosporins, and cimetidine may cause a falsely low serum level. Bacteria can break it down if it is kept at room temperature. A heavy meat diet can influence levels. It can't be used if the patient has a muscle wasting disease, must be corrected for body surface area, and is very sensitive to the sample not being collected correctly. The main source of error is improperly timed urine specimens. As the 24-hour starts, the patient should urinate in the toilet, then collect all urine in the next 24 hours. At the 24th hour, the urine should be collected and added to the container. The container should be refrigerated or kept on ice near the toilet, so it is best to do this when a patient will be at home for 24 hours. Estimated glomerular filtration rates. Estimated glomerular filtration rates can be added to the laboratory information system as a programmable formula to estimate clearance based on plasma levels of creatinine. This can be used for routine screening of patients in a metabolic profile or for monitoring patients who have been diagnosed with kidney disease or who are at risk for kidney disease. The National Kidney Disease Education Program recommends the MD-RD-IDMS-traceable formula. Indicators of glomerular filtration. Cystatin C and beta-2 microglobulin are substances that are produced at a constant level. Increases in either indicate a decreased glomerular filtration rate. Injecting radionucleotides and measuring their clearance is good for monitoring transplanted kidney function. Beta-2 microglobulin cannot be used in a patient with immunoglobulin disorders or malignancies. Clinical significance of GFR. What is the clinical significance of the glomerular filtration rate? It is not the detection of early renal disease. GFR is used to measure the extent of nephron damage in known cases of kidney disease. It is used to monitor treatment given to prevent further damage to nephrons. It can also be used to determine if certain medications should be used. Some medications will build up to toxic levels if the glomerular filtration rate is low. Tubular reabsorption is one of the first functions that is affected in kidney disease due to the complexities of the process. Because hydration level has a significant impact on the specific gravity of urine, the fluid intake must be controlled for this test to be effective. Currently, renal concentrating testing is performed after various periods of fluid deprivation, measuring urine and often serum osmolality. If a urine osmolality after fluid control is less than 800 millimosmoles, then a urine and serum sample are taken 2 hours later with no water. A urine to serum ratio of 3:1 or greater, or a urine osmolality of 800 milliosmoles or greater indicates normal tubular reabsorption. If the results are abnormal, you must rule out diabetes insipidus. If the results correct after injection of ADH, then they have diabetes insipidus. If it does not correct, then tubular damage is the cause. Osmolality can be measured by comparing either the freezing point or dew point of an unknown patient sample to a known value. If the sample has lipemic serum, lactic acid, and volatile substances such as ethanol in the specimen, this will interfere with the test. The clinical uses of osmolarity, including evaluating renal concentrating ability, monitoring renal disease, monitoring fluid and electrolyte therapy, establishing the differential diagnosis of hypernatremia and hyponatremia, and evaluating the secretion of and renal response to ADH. Another tubular reabsorption test is PAH, or p-aminohippuric acid test. This test uses a substance that is not naturally found in the plasma. It is a non-toxic substance that is loosely bound to plasma proteins. It is secreted in the proximal convoluted tubule, and the volume of plasma flowing through the kidneys determines the amount of PAH excreted in the urine. This must be administered by IV and would be conducted in a specialized laboratory. The titrable acidity and urinary ammonia test. The kidney's ability to produce an acid urine depends on the tubular secretion of hydrogen ions and ammonia production and secretion by the cells of the distal convoluted tubule. A urine pH, titrable acidity, and urinary ammonia measurements can be used to determine the defective function of the kidney to excrete hydrogen ions. Definitions and terms. Active transport. Movement of a substance across cell membranes into the bloodstream by electrochemical energy. Afferent arteriole. A small branch of the renal artery through which blood flows towards or into the glomerulus of the kidney. Aldosterone, a hormone that regulates reabsorption of sodium in the distal convoluted tubule. Antidiuretic hormone, or ADH, a hormone produced by the hypothalamus to regulate water reabsorption in the collecting duct. Beta-2 microglobulin, a subunit of the class 1 major compatibility antigens that enter the blood at a constant rate. Collecting duct, a part of the nephron where the final concentration of urine takes place through the reabsorption of water. Countercurrent mechanism, a selective urine concentration process in the ascending and descending loops of Henle. Creatinine, a substance formed by the breakdown of creatine during muscle metabolism. Cystatin C, a small protein produced at a constant rate by all nucleated cells. Distal convoluted tubule, part of the nephron between the ascending loop of Henle and the collecting duct, where the final concentration of urinary filtrate begins. Efferent arteriole, the small renal artery branch through which blood flows away from the glomerulus. Free water clearance, a test to determine the ability of the kidney to respond to the state of body hydration. Glomerular filtration barrier, structures of the wall of the glomerular capillaries that prevent the filtration of large molecules from the blood into the urine filtrate. Glomerular filtration rate, the volume of plasma that is filtered by the glomerulus in a specific time. Glomerulus, a tuft of capillary blood vessels located in Bowman's capsule where filtration occurs. Juxtamedullary apparatus, a specialized cell located on the afferent arteriole that regulates secretion of renin. Loops of Henle, the U-shaped part of the renal tubule that consists of a thin descending limb, a thick ascending limb. Maximal reabsorption capacity, TM, the maximum reabsorption ability for a solute by renal tubules. Nephron, a functional unit of the kidney that forms urine. Osmolality, the osmotic pressure of a solution expressed in milliosmoles per liter. It is only affected by the number of particles present. Osmotic gradient, the difference in the concentration of substances on either side of a membrane. Passive transport, movement of molecules across a membrane by diffusion because of a physical gradient. Peritubular capillaries, the capillaries that surround the renal tubules. Podocytes, epithelial cells of the inner lining of the Bowman's capsule that contain foot-like processes. Proximal convoluted tubule, the nearest tubule to the glomerulus where reabsorption of essential substances begins. Renal plasma flow, the volume of plasma flowing through the kidneys per minute. Renal threshold, plasma concentration of a substance at which active transport stops and increased amounts are excreted in the urine. Renin, proteolytic enzyme produced by the kidney that reacts with angiotensinogen to produce angiotensin to increase blood pressure. Renin-angiotensin-aldosterone system regulates flow of blood to and within the kidneys by responding to changes in blood pressure and plasma sodium concentration. Shield of negativity, the negative ions of the glomerular filtration barrier that prevent small proteins such as albumin from entering the urine filtrate. Titrable acidity, hydrogen ions in the urine that can be quantified by titration with a base to a pH of 7.4. Tubular reabsorption, substances move from the tubular filtrate into the blood by active or passive transport. Tubular secretion, the passage of substances from the blood in the peritubular capillaries to the tubular filtrate. Vasa recta, the network of capillaries that surrounds the loop of Henle. Questions. Question 1. What is the structural and functional unit of the kidney? A. Renal corpuscle. B. Nephron. C. Loop of Henle. D. Tubules. The answer is B. The nephron. Question 2. The renal corpuscle is made up of A. Tubule and collecting ducts. B. Cortical and juxtamedullary nephrons. C. Bowman's capsule and glomerulus. D. Podocytes and basal lamina. The answer is C. Bowman's capsule and the glomerulus make up a renal corpuscle. Question 3. What are the two types of nephrons? A. Cortical and juxtamedullary. B. Pedicile and juxtamedullary. C. Henle and collecting. D. Cortical and medullary. The answer is A. Cortical and juxtamedullary. Question 4. Correct flow of the blood through the nephron. A. Afferent, glomerulus, vasa recta, efferent. B. Efferent, glomerulus, afferent, peritubular. C. Afferent, glomerulus, efferent, peritubular. D. Peritubular, glomerulus, afferent, efferent. The answer is C. Afferent, arterial, glomerulus, efferent, arterial, and peritubular capillaries. Question 5. Glomerular filtration occurs due to the blank between the glomerulus and the filtrate in the Bowman's capsule. A. Sodium concentration differential. B. Osmotic concentration differential. C. Orthostatic pressure differential. Or D. Hydrostatic pressure differential. The answer is D. Hydrostatic pressure differential. Question 6. The renin-angiotensin-aldosterone system regulates blood flow to the kidneys to maintain A. Blood pressure and sodium levels. B. Blood pressure and acid-base balance. C. Bicarbonate and hydrogen ion concentrations. Or D. Creatinine and sodium levels. The answer is A. Blood pressure and sodium levels. Question 7. Water is reabsorbed by what process? A. Active transport. B. Passive transport. C. Hydrostatic transport. D. Creatinine clearance. The answer is B. Passive transport. Question 8. What effect does antidiuretic hormone have? A. Decreases urine volume. B. Decreases blood pressure. C. Increases urine volume. Or D. Decreases water absorption. The answer is A. It decreases urine volume. Question 9. To maintain acid-base balance in the tubules of the nephrons, A. Reabsorb chloride negative and secrete sodium positive. B. Secrete sodium positive and reabsorb chloride negative. Secrete bicarbonate negative and reabsorb hydrogen positive. Secrete hydrogen positive and reabsorb bicarbonate negative. The answer is D. Secrete hydrogen ions and reabsorb bicarbonate. Question 10. The test most used to measure glomerular filtration is A. Sodium clearance, B. Radionucleotide clearance, C. Creatinine clearance, D. Hydrogen ion clearance. The answer is C. Creatinine clearance. Question 11. The glomerular filtration rate is not used A. For detection of early renal disease, B. To measure extent of nephron damage in known cases of renal disease, C. To monitor effectiveness of treatment, or D. Feasibility of administering medications. Remember, the question is the glomerular filtration rate is not used for, and the answer is A. The glomerular filtration rate is not used for detection of early renal disease. It is used for the other three. Question 12. The National Kidney Disease Education Program recommends screening with an estimated GFR using A. Creatinine clearance on a 24-hour urine, beta-2 macroglobulin on a 24-hour urine, cystatin C on a 24-hour urine, or MDRD-IDMS-traceable formula. The answer is D. An estimated GFR using the MDRD-IDMS-traceable formula. So the lab information system will just calculate it based on blood results. What test is useful in evaluating renal concentrating ability? A. MDRD-IDMS, B. Creatinine clearance, C. Osmolality, or D. Cystatin clearance. And the answer is C. Osmolality. Okay, question 14. A urine pH blank and a urinary blank measurement can be used to determine the defective function of the kidney to excrete hydrogen ions. So A is titrable acidity / ammonia, B is osmolality / ammonia, C titrable acidity / hydrogen ion, D hydrogen ion / bicarbonate. The answer is A, titrable acidity and urinary ammonia. You can see fadavis.com to find the book Urinalysis and Body Fluids by Susan King Strassinger and Marjorie Schwab Di Lorenzo. Thank you for listening, and I hope that this helps you with your studies.